My name is Robert Rosenson. I'm a professor of medicine in the Division of Cardiology at the Icahn School of Medicine at Mount Sinai in New York. Today I spoke about HDL functionality. First of all, we have to get off this concept that HDL cholesterol is a surrogate measure of HDL. HDL cholesterol is just the cholesterol cargo in the HDL particles. In Mendelian randomization studies, HDL cholesterol is unrelated to coronary heart disease risk. The HDL particle and the concentration of the HDL particles are more important as risk predictors, particularly in statin-treated patients, as recently shown in the heart protection study in the Jupiter trials. Small HDL particles carry a different proteome and a lipidome that have very important functional features, such as the antioxidant, anti-inflammatory, and anti-infective properties associated with HDL. So looking at more detailed measures of HDL may be very important. But none of these compositional changes can substitute for functional measures of HDL. Most notable, the macrophage cholesterol efflux assay, studies of endothelial function, studies of vascular wall stability. These surrogate measures, which have been developed in multiple laboratories around the world, still need to be validated. Validation in terms of showing relationships with atherosclerosis, validation in terms of associations with cardiovascular disease. Sometimes the HDL particles can actually become dysfunctional. As shown in the work of Hazen and colleagues and Heineke and colleagues, free ApoA1 in the plaque can be modified by the effects of myeloperoxidase. And it turns the ApoA1 into a dysfunctional particle that actually increases inflammation by activating nuclear factor kappa B, and also interferes with macrophage cholesterol efflux by the ABCA1 transporter. So our concepts are changing, and before we start launching into more pharmaceutical tools to raise HDL cholesterol, we need to understand the biology a little bit better and focus on function versus dysfunction of those HDL particles. Thank you. Well, the Congress offers great opportunities to interact with individuals all over the world, people in different disciplines, and I've certainly had the opportunity to discuss research collaborations with some of the people that I've met for the first time at this meeting, and other individuals that I know to discuss research opportunities and some of the clinical dilemmas that we face in the context of this new scientific information. So I find it to be a great experience and a great opportunity for me. For the young individual, it affords the opportunity, this meeting, to meet, ask questions, discuss ideas, and then hopefully actually partner with people around the world to help foster development and get some of these ideas executed that will hopefully make importance for science and, more importantly, for patient care.